In lecture 11, we learned about hemophilia. Now, in this, it's a continuation of Mendelian disorder. We will be learning about sickle cell anemia, thalassemia, and phenylketonuria. So, let us start. Sickle cell anemia. It is a disorder related to the blood cells, in which the RBCs, which should be biconcave in shape, it does not remain so. So what happens? They become curved, okay, crescent shaped. They turn crescent shape. This is the disorder, sickle cell anemia, defective blood cells. It is an autosome linked disorder. That means, like the we had learned about hemophilia. Hemophilia is a sex-linked disorder. The defective gene is present in a sex chromosome, but in this case, the defective gene is located in one of the autosomes. Okay, hence it is called as autosome-linked disorder. Now, in this, what happens? As I mentioned, shape of RBC gets deformed to sickle-shaped or crescent-shaped under oxygen deficiency. So here we can see, okay, I will just zoom out and show here. So here you can see these are the normal shape of RBCs. These are the normal shape of RBCs, but under oxygen stress, if the person is getting less oxygen, the defective person is getting less oxygen, then his red blood cells are likely to take sickle shaped. This type of shape they will take, and consequently what will happen? They will clog the blood vessels. Okay, they will clog the blood vessels. That means they will block the blood vessels. So you can see here there is another picture. Okay, here this is the normal blood cells, and when they become sickle shaped, what will happen? They will block. So this way the blood vessels will be blocked, and the body will not, different parts of the body will not get a proper supply of blood. Okay, now how it happens? An abnormal hemoglobin leads to this condition. Okay, hemoglobin, which is the main component or constituent of RBCs, turns out to be abnormal. Why does it become abnormal? Due to a defective gene. Okay, due to a defective gene which transforms the, or which alters the shape of the hemoglobin, consequently the shape of RBC. Gene for normal hemoglobin. Gene for normal hemoglobin is referred to as HbA. Okay, we refer to it as HbA. Gene for sickle shaped HbS. We are representing it in this manner. So if a person has, and this is a recessive gene, okay, this is a recessive gene. So if a person has HbS gene, also HbA, that means the allele, the pair of genes, one is A and the other is S, then the person will be a carrier. But if the person has both HbS genes, okay, then that person will be suffering from sickle cell anemia. This is a recessive disorder, autosome linked recessive disorder. As it is a recessive trait, only homozygous will show the diseased phenotype, as I just now mentioned. Heterozygous forms will be the carriers. So parent carriers, HbA, HbS, okay, HbA, HbS. In the F1 generation, we will get, okay, we are likely to get an individual who will be with normal RBCs, okay. They have got the normal genes, both of them, the dominant genes. Then again, and two individuals might develop who will again become carriers, heterozygous forms, and one defective. One is likely to be defective. So who can have sickle cell anemia? A sickle cell anemic person will have both his parents heterozygous. Both his parents have to be, or her parents has to be heterozygous. Then only the person will be sickle cell anemic. Okay, otherwise, if one of the parent is a carrier, the other is normal, then the offsprings born will not be sickle cell anemic. But of course, they may again become carriers. Now, what is the molecular basis of sickle cell anemia? Why does it happen? Okay, what happens? HbA is the normal gene. Then what happens in sickle cell anemic people? Now, this defect was due to a mutation, a type of mutation. Normal hemoglobin is hemoglobin A, which changes to hemoglobin S. So, this hemoglobin S is the defective one. A person who has this hemoglobin S form, hemoglobin in this form, will be the affected individuals who will be showing the symptoms of the disease. Now, what happens? The 6th position amino acid of beta chain of hemoglobin A is glutamic acid. Now, what does it mean? Hemoglobin molecule is made up of globin chains, protein chains, alpha and beta chains, 2 each. Now, alpha chains are quite normal. Okay. Now, beta chain. Okay, here we are concerned with the beta chain. Alpha and beta makes up hemoglobin. The beta chain of hemoglobin A. Okay, beta chain of hemoglobin A, in it, the gene responsible for formation of this particular chain. Chain, when I am saying, it means a polypeptide. It means a protein. The gene is responsible for formation of protein. So, the gene which is forming this particular chain of hemoglobin A, okay, when everything is normal, then what will happen? The 6th position amino acid of the protein chain. Protein chains, they are made up of amino acids. So, we can number them: 1st, 2nd, 3rd, 4th, 5th, 6th. The 6th position is glutamic acid. A normal person will have glutamic acid in the 6th position of the beta chain forming hemoglobin. But sometimes what happens? Mutation, that is change in gene, takes place in the gene which will code for amino acid at the 6th position of hemoglobin. Mutation occurs in the gene. So what happens? In place of glutamic acid, it is replaced by another amino acid called as valine. And only such a simple, such a small, such a minute change, glutamic acid not present, replaced by valine. Why? Because the gene changed. Because there was a change in the gene underwent mutation, and it results in complete transformation of the hemoglobin. Complete transformation of hemoglobin S to hemoglobin A. Hemoglobin A, I'm extremely sorry. Hemoglobin A will change to hemoglobin S due to the transformation or due to the replacement of glutamic acid by valine. So here we have a picture representing it. Okay, now this is the DNA sequence. That means this is the part of the gene which codes for hemoglobin. Okay, which codes for hemoglobin. Here we can see this is a hemoglobin A that has been shown, which will give normal red blood cells. So here 1, 2, 3, 4, 5, 6th position. It will continue, but we are concerned only with the till the 6th position here. So the 6th position is occupied by glutamic acid in a normal hemoglobin beta chain. Okay, it's normal. So here glutamic acid, which is coded by the gene sequence. Here you can see the bases CTC and GAG. It codes normal. Now mutation takes place. Okay, point mutation or missense mutation takes place at this position. You can see here where it was CTC, GAG, it changed to CAC and GTG. T replaced by A replaced by T. So what happened? A change took place. Consequently, where glutamic acid should have taken position, it was replaced with valine. As it got replaced with valine, the effect or the consequence is so very drastic that in place of the normal blood cells, you get sickle shaped red blood cells. So this is how it changes, and of course this the change in shape of RBC will be observed under oxygen deficient conditions. Now again the same thing, it is represented here. It's a normal, okay, the DNA changed to sickle cell, okay, the DNA will undergo transcription to form RNA. RNA will form the protein. Okay, so here this is the normal one. So the RNA is normal, which forms glutamic acid. It brings glutamic acid. Here only the sixth position is shown. And when mutation occurs, here A replaced by T, T replaced by A. So what happens? Even the RNA changes. Consequently, in place of glutamic acid, we get valine. Again the same thing, okay, for better understanding. Again we have partial DNA sequence of the beta globin gene. So this is the partial part that has been shown here, okay. So here missense, the mutation occurred, point mutation also it is called as, or missense mutation. A replaced by T, T replaced by A. So the RNA that was formed here, the normal one is GAG, but due to mutation the RNA formed will be GUG. GUG codes for valine, GAG codes for glutamic acid, normal. So here it is normal, but here what happens? Valine is formed, due to which the RBC is deformed. So now why all this, why the change in RBC occurred? It occurred due to change in the gene, and this gene sequence, like if once it had occurred, it is present in the person, it's likely to be, or it will be transmitted, or it will be transferred to the next generation. So it runs down in the family. This defective gene will run down in the family, and coincidentally it happens both the parents have one gene with the defect, so they are likely to produce an offspring who will be having the in whom the homozygous form will appear, and the person will be suffering from sickle cell anemia. Now we will move on to the next type of disease, quite in similar type. Okay, the inheritance pattern is quite similar. It is phenylketonuria. This is also an autosomal recessive disorder. Why does it happen? It happens due to lack of enzyme. The person who is suffering from phenylketonuria, in him or her body, the enzyme phenylalanine will not be produced. Okay, which in a normal person, it will help in conversion of amino acid phenylalanine, which we get by consuming proteins. Okay, when we consume protein, we get phenylalanine in our diet. Okay, along with the diet, it enters the body and it gets converted to tyrosine in the liver. Okay, and tyrosine will further undergo different metabolic pathways. Now, okay, so a person who is suffering from phenylketonuria, no, this enzyme will not be produced. So consequently, the conversion of phenylalanine will not take place in the body. Resultant phenylalanine will get deposited in the body, will remain in the body, and get converted to phenylpyruvic acid, which is quite harmful for the body. It will affect the brain, result in mental disorders. Inheritance pattern is quite similar to sickle cell anemia. Okay, now we move on to the next type of disorder, thalassemia. Now what is thalassemia? Thalassemia is also an autosomal recessive disorder. Even in this, what happens? Due to mutation or deletion of genes controlling formation of globin chains, alpha and beta. Okay, now why does it appear? This is also an autosomal recessive disorder, means both the parents have to be carriers, then only the child is likely to be affected. Okay, now why does it happen? It happens also due to mutation or deletion of genes. What genes? Which is controlling the formation of globin chains. Just now I had mentioned that in sickle cell anemia, the hemoglobin, it is made up of alpha and beta chains. Okay, and in the sickle cell anemia, one of the beta chains is affected. Now in thalassemia, what happens? Both alpha and beta can be affected. it is referred to as a quantitative problem. Quantitative problem means there is no formation of globin chain. The number of globin chain which is supposed to be formed will not be formed. So it is quantitative. But in sickle cell anemia, it is qualitative. The quality of globin chain is affected. What happens? The person is anemic. Whoever has thalassemia, the person will be anemic. There are two types of thalassemia, alpha and beta. Now, what is thalassemia? Okay, the same thing again. Genetic blood disorder resulting in a mutation or deletion of genes. Normal hemoglobin is composed of 2 alpha, 2 beta. Mutation in a given globin chain can cause decrease in production of the globin, as I mentioned just now. And then what happens? Aggregates become oxidized, damage the cell membrane, leading either to hemolysis, ineffective erythropoiesis, or both. That means what happens? The structure of RBC will not be normal, because if the components that is making up RBC, they are not completely formed, then how can its structure be concrete? What will happen? The RBC it becomes labile. That means it will very easily break down before it matures. Normal RBC, they should last for 120 days. But the persons who suffer from thalassemia, in their body, the RBCs will never last for 120 days, but it will break down quite before that, depending upon the severity, how many globin chains are affected, depending upon that, it will break down. So ineffective erythropoiesis, that means ineffective breaking down of RBCs. The types alpha and beta. Again, there are subtypes. An alpha type again can be some silent carriers, some can be traits, some can be hemoglobin as intermediate, major, hemoglobin constant, spring. These are the different types of alpha types. Beta types, again, it can be 3 types. We will see now how it happens. Before going into it, let us little bit about the RBC. RBC, as I had been mentioning, it is made up of globin chains, hemoglobin, heme, iron, globin, protein chain. So 4 protein, so here you can see these are the protein chains, the 4 protein chains. Alpha, okay, this is the alpha chain, okay, beta chain, here beta chain and the alpha chain. The red and purple are the alpha chains, green and pink are the beta chain. Each of them has one iron heme group. That is why hemoglobin. Now, the people who suffer from thalassemia, in them the formation of this alpha chains will be affected, or the beta chains will be affected, or in some very severe cases, the alpha as well as beta chain might be affected. So, what happens? The structure of RBC gets distorted. So here now let us go further into it. Now chromosome number 11, okay, and chromosome number 16 in our body, okay, the pair of chromosomes, they carry the genes which is responsible for formation of beta globin, okay, beta globin chain of hemoglobin. They have the gene, chromosome number 11. Chromosome number 16 has the alpha globin gene. That means they have the genes which will help in formation of the alpha chain of hemoglobin. Okay, so chromosome number 11 and 16. Now here we can see chromosome number 16, that is which forms the alpha, okay, which forms the alpha chain, globin chain. Okay, here these are the genes, okay, a pair and a pair and a pair. So this is one of chromosome number 16, the second of chromosome number 16. Okay, that means what happens? There are 4 genes, okay, or we can say 2 pairs of genes controlling the formation of alpha globin chain. So each of them, okay, responsible for 25% of the globin, alpha globin, one of the gene. Second gene 25% of the alpha globin, third one 25%, and the fourth one 25%. So if one of them is defective, then what will happen? Only 75% of alpha chain will be formed. So it's not complete. Okay, it's the chain formation will not be complete. So obviously, if it is not complete, then it's likely to break down easily if alpha gene is defective. And in chromosome number 11, what happens? We are talking about beta. Okay, we are mainly focusing on beta globin chains. Only one pair is present of beta gene. But of course, though it's like little beyond our discussion at your level, but still then just to mention, also in chromosome number 11, there are other two pairs of genes which helps in formation of the RBC, which are referred to as delta and gamma genes. Okay, delta and gamma genes. Now they will help in formation of the beta chain together. But we put more emphasis or more focus on the beta genes located in chromosome number 11. So here, if one of the beta gene is defective, then definitely the beta globin chain will be defective. If both of them are defective, then a major portion of the beta globin chain will be defective. Likewise, suppose 3 defective alpha genes, then a major portion of the alpha globin chain will be defective. So that is how it happens. So here again, chromosome number 11, gene defects. When a mutation on one of the 2 beta globin genes, a carrier is formed in lower protein production but not enough hemoglobin. Okay, so as I had been mentioning, okay, without a mutation, enough hemoglobin. Okay, no mutation, it's quite normal. Okay, the chromosome number 11 which is carrying, okay, it has normal chromosome number 11, that is the beta globin. Okay, quite normal. Suppose one gene is affected, so what will happen? Beta thalassemia carrier but less hemoglobin, slightly anemic. Both of them are affected. Okay, beta thalassemia major, then this is a major, because a major portion of the globin chain will not be formed. So HBB gene on chromosome number 11, it is named as HBB. Okay, hemoglobin beta gene on chromosome 11. Then chromosome number 16. Okay, what happens in chromosome number 16? As I told you, there are 4 alpha genes. Okay, 4 alpha genes or 2 pairs. If one is defective, okay, here, or 3 may be defective, okay, then it is a major one. This is a silent carrier. Here it is normal. None of them are affected. Okay, and if this is the case, say 2 are defective. In this manner, 2 are defective. That means 1 each from both the pairs or 2 from the same pair. Whatever be the case, then we will say these are traits. Alpha thalassemia trait this person will be having. And the genes, they are named as HbA1 and HbA2. 4 alleles. HbA1, 1 pair. HbA2, another pair. So 4 alleles. So now the thalassemic disease, the severity of the disease will depend upon how many of the genes are defective. If suppose it happens that the HBB gene on 11 is also defective, then HBA in 16 is also defective, then this is a very, very severe case. If only 1, then it is a minor case. If 2, then we can call it major or we can call it trait. Okay, 3, then it is also a major trait. So here the 2 types, alpha and beta thalassemia. Alpha thalassemia, defective alpha chain, summing up everything now. Alpha, defective alpha chain, genes responsible HbA1, HbA2, located on chromosome 16, which has 4 alleles, 2 each. Okay, 2 each. Then if 1 defective allele, it will be a carrier. Alpha minor, if 2 defective alleles. Fetal death, if 3 are affected, then it may lead to fetal death. That means the fetus may die before birth. Beta thalassemia, decreased beta globin chain. Why does it happen? Due to defective HBB, that means hemoglobin beta gene on chromosome number 11. If 1, then we will call it minor. Major, then 2 defective. If both of them are defective, then we will call it major. Okay, so this is about thalassemia, and these are the different types of Mendelian disorders that we have to study as per our curriculum. So what we learned about hemophilia, we learned about sickle cell anemia, we learned about phenylketonuria, and we learned about thalassemia.